Good afternoon, everyone. Let's get started. Today, we're going to be doing lecture 3, which is the immune system. It has quite a bit of information in this lecture, so make sure you do ask questions if you are confused at any point of the tutorial. All right, first question, list 4 characteristics of innate immunity. All right, so if you think about immunity as a general system, right? So immunity, it is separated into 2 branches. We have the innate immunity, and we have the adaptive or acquired immunity. Okay, so these are the 2 major branches. The innate is the one that you are born with. The adaptive and the acquired is the one that will change throughout your lifetime. You actually have to be exposed to the pathogen before you have the ability to fight them off. That's why it's acquired, and it's adaptive in the sense that every time you encounter a new pathogen, you kind of add that to your immune system. And throughout the course of your life, the pathogen library, so to speak, that is within your immune memory is going to grow. So let's talk about the innate immunity first. The 4 characteristics of the innate immunity include: number 1, it is non-specific. It is not designed to fight off one particular pathogen, right? It's kind of like a catch-all net. It's a broad defense system that would work against a wide range of pathogens. But of course, if you are infected with some of the stronger pathogens, more difficult ones to fight off, then innate immunity would not be sufficient. Okay. Another characteristic is that it will work immediately. There is no lag time. It would be able to start fighting the pathogens right away. And that's the second characteristic. What's the third one? Does anybody remember? What's the third one? Third one is it is inherited. Right? You're born with it. The moment you come into this world, you are ready to fight pathogens because the innate immunity will be ready to go. And lastly, it has no immune memory. Right? It doesn't remember what you have thought of, and it's not a long-lasting type of immunity. Okay, so there you have it for characteristics. Now, this table talks about the various ways that innate immunity has to deal with infection and pathogens. Okay, the 4 various mechanisms. The first one is barrier to entries. Okay, so I'm gonna give a little description of it, and then I'm going to give you some examples. So I'll put the purpose in blue, and I will do the description. I'll do the description in another color. Okay, so the purpose, right? Purpose of the barriers is basically to prevent. This study is different format. What do you mean this study? Oh, you mean the study guide from the book, from the one that you purchased? Is that what you mean? Yeah, sorry. Again, there are subtle differences, right? But anything that I do add are usually just tables that are really easy to copy on a separate piece of paper. So again, I apologize for that. So barriers to entry. Oh, you download and it's different? Really? Let me see what's going on here. Give me a second. Let me pause the recording first. Pause. Hello, for people who are watching this from the internet, we actually had the wrong study guide, but now it's fixed. So if you didn't know what was going on for the first 5 minutes, Now it should make sense. So let's try this again. So immunity, right? We have the innate immunity and the adaptive immunity, right? Okay, so let's talk about the response time. Okay, so for innate immunity, I already told you it is going to be immediate, right? Immediate. Right? Whereas adaptive immunity is going to have a delay. Okay, there is a wait period. Okay, so not immediate. It's basically the same information, but in a table format. Specificity, non-specific. Okay, non-specific, not designed to fight any particular pathogen, whereas for adaptive immunity, it is specific. Right, you're gonna have antibodies to fight chickenpox virus, particularly. You're gonna have antibodies that fight hepatitis specifically. How antigens are detected for innate immunity? It relies on PAMS, right? PAMS, okay, which is pathogen associated. Excuse me. Pathogen associated molecular patterns. Okay. And for adaptive immunity, to detect antigens, we rely on gene shuffling. Okay, so by shuffling the gene, you're going to be able to create unique receptors that are on your white cells, and those receptors will then be able to detect foreign antigens. Okay. Type of cells involved in innate immunity. So we have things like, macrophages, right? Phagocytes, NK cells. Okay. These are all cells that you will find as part of the innate immunity. What about adaptive immunity? What kind of cells do we have in the adaptive immunity? Help me out here.
B cell, T cell.
That's right, the T cells and the B cells. Okay. Innate immune memory, none. Okay. And then for the adaptive immunity, it is long lasting. Okay. If you fought off chickenpox when you were young, you will have a copy of that B cells and T cells circulating in your system right now. That's why you don't get it again. Okay, so there you have it. Hopefully that makes more sense. Sorry about the confusion again. All right, let's go to question number 2, which is the mechanism for innate immunity. So again, I will do the purpose in blue, and then I'll do the other one and then the color later. Okay, so what's the purpose of for the barriers to entry? So basically, we prevent pathogens from entering the body, right? Or if they do enter, we can prevent them from colonizing on bodily surfaces. Okay, we don't want them to attach to our respiratory tract, for example. We want to get rid of them. Okay. So there are different types of physical barriers. Does anybody have an example for me? What are the two types of physical barrier? Mohammed has a question. Is adaptive better than innate? Well, it's better in the sense that it's more tailored. So for the really difficult pathogens, for the more like dangerous pathogens, like COVID, for example, right? Innate immunity will not be able to deal with it. Then you need adaptive immunity. But you know, for everyday stuff, like if you go out, have a walk, you breathe in a lot of pathogens, then innate immunity is the better one, because it's always ready to go, and it's able to clear the pathogens, like, you know, relatively quickly. So it really depends on what situation we're talking. Okay. Prashel said skin, mucus, and membrane. Yep, that would be example of physical barriers, right? Physical barriers. Okay, so we have skin, intact skin, right? No breaks in it. Mucous membrane. So like the inside of the mouth, for example, right? Ciliated cells. Ciliated cells. Right? And also normal flora. Normal flora are these good bacteria, good microorganisms that lives on us, in us, right, within our intestinal tract, on our skin, and they take up so much space that there will be no rooms left for the bad ones. Okay, so normal flora, we have a symbiotic relationship with them, right? We have basically we benefit each other. We give them a place to live, we feed them, and in turns they protect us. Okay, it's a mutually benefit situation. Other than physical barriers, what's the other category?
Chemical.
That's right. The other one is the chemical barrier. Thank you. So physical and chemical barriers. Chemical barriers would be things like tears, right? Actually, I think I used the word lysozyme, which is a type of enzyme that will break down the bacteria. So lysozymes in tears, sweat, right, and saliva. Someone says low pH. That's right. So low pH in the stomach, for example. Okay. So there you have it. That is the barriers to entry. The best defense is to, you know, not let them come in at all. But if they do come in, we can sometimes slow down the infection by mounting an inflammatory response. So help me out here. What cell is responsible for creating the inflammation? Does anybody remember? What cells is responsible for mounting the inflammation? That's right. So inflammation can be triggered by mast cells, right? In tissue. Okay, so they release histamine, and the purpose of inflammation is to recruit more white blood cells and plasma protein to the site of infection. Site of infection. Okay, so what kind of WBCs are we talking about? Well, we can recruit more neutrophils, right, and more macrophage. These are both examples of phagocytes. So you come and eat up things that are not supposed to be there. And then for plasma proteins, we would want to have things like some generic antibody. Sometimes we might need to have some clotting factors. We might have some complement proteins. Okay, so these are both important. And that's what inflammation does. All right. So inflammation is triggered by the release of, what's the chemical that causes all these to happen?
Histamine.
Histamine, that's right. Thank you. So the mast cells will release the histamine, which will then, number one, it will promote vasodilation. Okay, make the blood vessels bigger so more blood can flow there. And number two, it will increase permeability. Okay, so make the vessels more leaky, right? So all these white cells can come and then they can exit the tissue and they could clear the pathogens from your tissue. Okay, so more on inflammation later on. Fever is another mechanism used by innate immunity. Okay, and the purpose of fever is basically to increase body temperature. Right, which allows a bunch of things to happen. It will, for example, increase your metabolism so you can make more defense protein. It will slow down the activity of bacteria, so on and so forth, right? So that's the purpose, increase body temperature. And what it does is there are these chemicals called pyrogens, things that can trigger a fever. Pyrogens such as prostaglandin, which you learn in the lecture, will act on the hypothalamus, which is part of the brain that controls many things, including body temperature. It will act in the hypothalamus to increase body temperature. Okay, so this thing right here, the hypothalamus, which we'll learn later in lecture 6, I think, that's called a master gland. Master gland. Many things that are that happens automatically in the body are controlled by that. So, hunger, thirst, sleep-wake cycle, body temperature, that's controlled by the hypothalamus. Are we okay here? Any questions so far? All right. Non-specific phagocytes. Okay, non-specific phagocytes. So these include things that such as neutrophils and macrophage. And the purpose of the non-specific phagocytes is to engulf, right? Engulf, to eat up, to engulf any cells that display foreign antigen. Okay, so what happens is they could recognize these foreign antigens, such as PAMs, right? Pathogen-associated molecular patterns. These are a group of molecules that are found on bacterial cells, for example, and your white cells will recognize them immediately. Okay. Another thing that's important to know for these non-specific phagocytes, I should put an example here. So like macrophage, neutrophils, right? Okay. Another purpose for non-specific phagocytes is that it serves as a link. Okay, it's a link between innate and adaptive immunity. Okay, through a process called antigen presentation. All right, so just to help you refresh your memory on what that means. So let's say you are infected with a red bacteria, and then here we have a macrophage. Okay, macrophage. Okay, so here this is bacterium. Okay, so what's going to happen is the macrophage is going to eat up the bacteria, right, using phagocytosis, right, which basically means it will come and engulf the bacteria. I hope that makes sense so far. Right. Just put a few more here. Right. And then what happens is it will do something called antigen processing, which basically means it will break down the bacteria and it will take the bacterial antigens, the molecules that are on the surface of the bacteria, it will put it on its own surface. Okay, so these are bits and pieces of bacteria, basically. Okay, and those are the bacterial antigen. Okay. What it does next is it will do antigen presentation. Right? If you watch the lecture recording, you will know that it will actually show it to some of the T cells and trigger them to differentiate, right? To clone themselves. Okay, so this whole process is necessary for activating the adaptive immunity. And so just to clarify, the antigen processing is the ability to break down the bacteria and put some of the bacterial antigens on the surface, right? And then to show it to a T cell, that is called antigen presentation. So not all cells is capable of doing this. Macrophage can do it. So we have another name for macrophage. Okay, they are called antigen presenting cells, APC. All right, so neutrophil is not an APC. Macrophage is an APC. I'm going to take a pause here and see if you guys have any question. Anybody have any question? Okay, well, let me know when you do have any. Next, we have the NK cells. What does NK cell stands for? Anybody remember? NK cells.
Natural killer cell.
That's right. These are the natural killer cell. It's kind of a violent name. And the purpose of the natural killer cells is to kill infected cells. Any cells that has been infected with a virus, for example, the NK cells will recognize them and kill them. And also cancerous cells. And the way that it does it, the description, right? The way that it does it is the NK cells is able to, NK cells can trigger apoptosis in target cells. Okay. Apoptosis is programmed cell death. So these NK cells will bind to the target. Maybe the target is an infected cell, maybe the target is a cancerous cell, and then it will cause that cell to basically kill itself off. Okay, so that's apoptosis. Right. Programmed cell death. Like inside each cell, there is like a button, right, so to speak. That's like self-destruct button. Right? So basically, the natural killer cell will cause these target cells to press that button and destroy itself. Okay? And that's a good thing, right? We want them to destroy themselves when A, it's an infected cell, or B, it's a cancerous cell. All right, next one, complement proteins. So these complement proteins, they are made in the liver, and there are many different complement proteins. They can be turned on or off, right? When you're not fighting an infection, they are typically off, right? And when you turn them on, their purpose is to enhance other immune response. Okay. In the lecture, I gave you three ways of how complement proteins can enhance other immune responses. Does anybody want to help me out here?
Recruit white BC.
That's right. So they can leave behind chemical trails for white cells to follow, and that allows them to recruit WBC to the site of infection, right? If you think about a white cell coming out from the bone marrow, freshly minted, doesn't know where to go, but with the chemical trails left behind by the complement proteins, they are able to find their way to the site of infection quickly. Hammer said coating target cells. That's right. They could tag pathogens to enhance phagocytosis. Right? By tagging the pathogens, it highlights them. It puts a red flag on them so the white cells can see them easily. And the last one, what's the last one? Well, fever is another mechanism that we talked about, but not specifically related to complement.
Membrane attack complexes.
That's right. They could form membrane attack complexes. Okay, membrane. Let's just spell it out. Membrane attack complexes. Okay, or MACS. Right, that will punch holes in bacterial cell wall. Okay, that's it. Those are your complement proteins. All right, do we have any poll questions? Let's do some poll questions here. Go ahead, give it a try. Yep, physical barrier will be ciliated cells, right? Okay, the stomach acid is low pH, that's chemical. Lysozyme is found in tears, sweats, and saliva. That's chemical, and tears also chemical. Let's try another one here. You guys should maybe like take a picture of these poll questions, right? So you can look back to them later on. I mean, you can always check the recordings as well, but probably is easier if you just snap a picture of the poll questions, because some of the questions on the quiz are just like the poll questions, right? I don't know if you realize that. All right, anyone else? So, histamine is going to, number one, cause vasodilation. So the blood flow, the vessel gets bigger, and it will increase blood flow. So that's not true. Fever has to do with the prostaglandin, not with histamine. And number two, the second thing that histamine will do is it will increase vessel permeability. It will make them more leaky, right? So if it's more leaky, then the white cells will be able to go from the blood to the tissue, not the other way around, right? Okay, and with all the liquid oozing out of your blood vessels, it will cause localized swelling. So that is the answer. All right. So thank you for participating. Let's do another one right here. PAMS. All right, I'm going to end the poll. These are molecular patterns that are found on pathogens, right? As the name implies, pathogen associated, right? So the answer is C. The phagocytes, such as white cells and such as macrophage, they will recognize the PAMs on the pathogens and therefore know what to engulf. Okay, let's do one more. we just talked about this one, right? The membrane attack complexes are formed by complement proteins, which would then punch holes in the walls of the pathogens, of the bacteria. Sorry. Okay, that's that. Moving on. Examples of physical barriers. We did this already, right? So, and we also did examples of chemical barriers. Let's just quickly write them down. We have skin, we have mucous membrane, and we also have normal flora, for example. Examples of chemical barriers: low pH, right? Lysozymes. List 2 effects of histamine on blood vessels. We mentioned this multiple times already. Number 1, vasodilation, right? Which would basically increase blood flow. And the side effects of increased blood flow is there's going to be some warmth and redness. Second effect of histamine on blood vessel is increased permeability. Which will allow the WBC and plasma proteins to leave blood vessel and enter surrounding tissues. Okay, and because you have all these things leaking out of your blood vessels, the side effect is that it will be there will be some swelling, and with swelling, there are more pressure on the nerve endings, and it will be painful. And so these are the 4 characteristics of inflammation. Okay, so these are characteristics. Okay, the kind of pain associated with inflammation is usually described as throbbing pain. If you feel like it's pulsating. If you have ever had a cut in the finger, like when you're sleeping at night, you can feel almost the finger, the heartbeat in the finger, right? That pulsating pain is characteristics of inflammation. Are we still together, everybody? Just checking in, see if anyone have any question. 3 immune functions of complement proteins. Okay, see above. We did that already. Right. List 3 effects of fever that will enhance immune defense. It will, for example, increase cellular activity. Okay, so e.g., faster phagocytosis. Right. It will reduce pathogen activities. Right. So, e.g., slow down bacterial division. Okay. It will also increase chemical reactions. So, such as complement protein activation. Remember, I said the complement proteins can have an on and an off form, right? So to turn them on or turn them off, it's actually chemical reaction. And you know, when we have a higher body temperature, those reactions tend to occur faster. Next, list 4 types of T cells effector and state their main function. So let's backtrack a little bit and revisit the concept of antigen presentation, right? So earlier I was telling you that the macrophage here, right, which is our antigen presenting cell, our APC, right? It has all these bacterial antigens on it. Okay, and so what happens is in your thymus, for example, your thymus is right here in front of the heart, you will have a population, a pool of naive T cells, right? So these are naive T cells. Okay, naive T cells. So these cells have not been activated yet, and each of them has a different receptor. So some of them will have like a blue receptor, right? Maybe a red receptor, green receptor, and then you might have one with pink receptor or something. And each of these receptors are actually proteins, right? So the T cell receptors are proteins. Okay, and we generate them using genetic shuffling. All right, so like I don't know if I talked about this in the recordings, but very quickly, you remember last semester that you have introns and exons. You guys remember that? Have you talked about introns and exons? Introns, exons?
I don't think no.
Sorry? Did
no,
you?
I don't think so.
No
No.
introns, no exons. Oh boy. Okay, well, you guys know like transcription, right? Transcription to translation. You guys, how about that? You talk about transcription, translation?
Yes.
Yes. Okay. So like, you know, you have your DNA and then you're going to do transcription to form RNA, right? So that's transcription. Okay. So when you copy the DNA, what happens is you actually copy a bunch of stuff. Okay. The gene, right? The gene. You copy the whole gene. But it turns out the gene has some junk DNA in it. Okay. So we have what we call exons. Exons are the actual coding portion. And then we have introns, which are the junk, the non-coding DNA. Right? So like we might have an exon number 1 here. We will have an intron here, exon number 2, intron number 2, exon number 3, right? Okay. So what you must do is you actually have to cut out all the junk DNA and connect the exons together. Is everybody with me about what exon introns are, right? So basically when you copy a section of a DNA, when you copy the gene in the form of RNA, that gene, not the whole thing, is useful for making protein. Okay, there are some introns in it. There are some non-coding junk DNA in it, and we must remove those before we can use the RNA to make proteins. Okay? Now, what happens is you have a set of genes that are dedicated for making these T cell receptors. Okay? So over here, right, this gene, in the simplest sense, I will remove intron number 1 and remove intron number 2, and I can join exon 1, exon 2, exon 3 together, and that creates one protein for me, right? And give me a thumbs up in the Zoom icon if you know what I'm talking about. I just want to make sure that you're okay with this. Right? So most of the time that's it. Okay, you just gotta make one protein, and you know life is good and everything is well. However, when it comes to the T cell receptor, you can do what's called gene shuffling. And gene shuffling means right? Instead of just joining 1, 2, and 3, right? It's okay to just not include 3. You just join 1 and 2 together. Or you can just do 2 and 3 together, or 1 and 3 together. So there are different combinations that you can create, right? So with just 3 exons, you can generate up to like 7 different combinations. Watch me. So like you could do like 1, 2, 3 together. That's one protein. 1, 2, that's 1. 1, 3, that's another one. You can do 2, 3, right? That's another one. You can go what am I missing anything here? 1, 2, 3, 4. You can go 1, 3, 2, 3. Oh, I think that's it. Sorry, you can go like 1 or 2 or just 3, right? So 1, 2, 3, 4, 5, 6, 7. There are 7 possible combinations just with 3 exons, right? So it turns out the genes that are dedicated for T cell receptors, there are like hundreds of exons. So by combining these different exons, every time you make the receptor, you can create a different receptor every time, basically. Okay, and that's what it is. In your thymus at any given time, you will have a population, certainly more than 4. Okay, you're gonna have a population of T cells. Each one of them has a unique T cell receptors. Okay, and they are different shape. Right? So I just use different color to represent the different shape, but in reality it's just different shapes. Okay, and so you will have the blue one, red one, orange one, green one, pink one, and a bunch of ones. All right, so what happens is, because we are infected with the red bacteria, the phagocyte's job is to go and present it to one of these naive T cells and see if there are any suitable receptors that already exist in that pool, right? So it will show it to the blue one, and it won't fit, so that's not going to be chosen. That's not going to work. That's not going to work. And if you are lucky, you would already have a naive T cell with the right receptor ready to be selected. Sometimes just by chance we don't have it yet, then you have to wait. Right? Your body constantly make new naive T cells, right? They constantly shuffle the genes to create new receptors. Okay, and so if you find that right one, it will undergo clonal expansion. Clonal expansion. It will make many, many, many copies of itself. Right? All of them would have the red receptor, because that's the one that you need, right? Okay, and after clonal expansion, this population will differentiate into effectors, right? That specializes in doing certain things, and that's what this question is asking, right? 4 effectors of T cells. Do we have any question about this process before I fill in the chart for you over there? Really? Everybody's good? Okay, well, maybe it is easier than I thought. So the effectors, what can those T cells become, right? They could become, for example, a killer T cell. A killer T cell, not to be confused with the natural killer cell, right? So these are killer T cells. Okay. Also known as cytotoxic T cell. Both is okay. Okay, that's the same thing over there. And as the name implies, they kill cells, and they are toxic to cells. Right? Their job, their function is to kill infected cells. Okay. By creating holes in them. Okay, they use the chemical, the porphyrin, that will put holes in them. So that's in your PowerPoint slide. What's another effector? Anybody remember from the lesson? Another effector.
Memory cell.
Okay, yeah, I hear memory cell. I'm gonna put it at the bottom here. Memory Memory T cells. And someone said something else?
Suppressor.
Yep. So memory T cell, suppressor T cell, and just one more. Yep, Tina said helper T cell. Yep. That's right. Okay, so that helper T cell, it will go activate naive B cell. Okay. And the B cell will then undergo clonal expansion. The suppressor T cell, the function is to stop immune activities when infection is cleared. You don't want your white cells to keep on cloning themselves when the infection is gone, right? Uncontrolled proliferation of white cells, it's basically like leukemia, right? So we don't want that. Memory T cells, it will stay in the body. In case of future infection by the same thing, right? Okay, by the same bacterium or by the same virus. All right. So like I said, for every diseases that you have been vaccinated for or that you have been exposed to and recovered in your blood right now, in your system, there will be a circulating memory T cells, right? Designed specifically for that particular infection. Okay. So when the thing comes again, you will be able to fight it off much quicker, right? The symptoms will be milder compared to previous exposure. Okay. So the T cell, the branches of immunity that are carried out by the T cells, we call this the cell-mediated immunity. Okay. All right, so for the B cell, right, the helper T cell will select a B cell, just like the APC will select the T cell, right? So the B cell with the correct receptor, the red one again, will be selected by the helper T cell. It will in turn undergo clonal expansion, and then it will differentiate. Right? So this kind of immunity, it is called antibody mediated immunity. Okay. Or humoral immunity. In case you read about it in a different name, they mean the same thing. Right? So we have two types of effectors for our B cells. They could become plasma cell, or they could become memory cell, memory B cell. Okay, so plasma cell, the function of plasma cell is to mass produce antibodies. And the memory B cell, their job is to stay in the body. Okay. So that's that. All right, let's do some of these long answer questions. We did inflammation already. I explained the role of APC. Okay. Explain how antibody works as part of your adaptive immunity. Let's do that. The antibody works by, well, first of all, let's just quickly review what antibodies are. Antibodies are Y-shaped proteins, right? Okay, Y-shaped, capital Y, Y-shaped proteins, right? And for the antibodies, we have a constant region, which are the same for all antibodies. And then we have a variable region. That's the part that binds to the antigen, that binds to the foreign antigen. Okay, so how does antibody work? Well, a couple ways. Number one, it could form an immune complex. Okay, which would then cause things to clump together. We are familiar with this concept from last week, right? When the anti-A antibody binds to a red cell that has protein A on it, it causes it to clump together, and then it causes them to burst, right? Hemolysis. So similar things can happen if you have a bunch of bacteria. The antibodies can bind to them and clump them together to form these immune complex. And the bigger they are, the easier it is to swallow them, right? Number two, they could tag pathogens, right? Kind of the same job as complement proteins. After tagging them, you can highlight them, making it easier for phagocytosis to take place. After tagging them, you can activate complement, right? So the two kind of feedback on each other. What's another one? Anybody remember? What's another function? Something to do with the virus. Anybody remember? That's okay. It could neutralize toxins and virus. Okay, viruses. Toxins are these harmful substances that are produced by bacteria. If you think about botulism, right? Botulism, you might have heard of Botox, right? Botox are these things that people can, inject underneath their skin to make their skin wrinkle-free, right? But that's basically a bacterial toxin, right? That can cause food poisoning and people could die from it. So the botulism, the bacteria that causes botulism, they produce these toxins, right? That causes muscle paralysis. It's not the bacteria themselves that causes the disease, but the toxin that they produce. So even if you cook your food completely and you kill off the bacteria, the toxin might still remain in the food and still cause you to have food poisoning. And so to neutralize these toxins, to make them harmless, the antibodies can bind to them. Okay. Same thing for viruses. Okay, if an antibody binds the virus before they enter the cell, it basically blocks their entry. Okay, and last one is the ability to activate complement proteins. Okay, so there you have it for 4 ways of how antibody works. Briefly describe the function of various T cell effectors. We did that already. If they seem a little bit repetitive, that's because these study guides were created for like in-person testing, right? In class learning. So like on the test, sometimes I would ask the question with a chart format. Sometimes I would ask it in like, written response format. So that's why I have both type of questions here. Do you guys want me to explain the active passive immunity again, and then active passive immunization? Yeah, that usually is a little bit confusing. Maybe
Yes.
yeah. Okay,
Yeah.
let's go through that one. Okay. I'll do it at the end, so when I pose it, it will be there. Okay, so again, we have immunity, right? Immunity, the ability to fight off pathogens at the top here. And we learned that immunity is separated into innate, right? And adaptive or acquire. So under adaptive, we can separate it into two categories. Right? It could be active or passive. So in active adaptive immunity, active means host needs to make antibodies. Okay, the host is the person who has the infection. Okay, so like you are the host, right? So you have to make your own antibodies, and to make the antibodies, that means you have to go through the whole selection process that I talked about, right? You got to find the right naive T cell. You have to wait for the clonal expansion. You have to wait for the differentiation to become effectors, and so that takes a bit of time. So it is not immediate. That's why when you get sick, right? When you get infected with something, you will feel not well for a couple of days, right? Sometimes for like a whole week or 2 weeks. During that whole time, your body is trying to make the antibodies, right? So it's not an immediate situation, not immediate protection. Okay, so some diseases are very deadly because they are able to cause a lot of harm during that time that you are trying to create the antibodies, right? That's why vaccinations are important. It allows your body to make those antibodies ahead of time, so when the real thing comes, you will already have the immunity for it, right? So that is not immediate, but once you got your effector cells, once you have your T cells differentiating, you will create the memory cells, and those memory cells will allow the protection to be long lasting. All right, so that's active immunity. Passive immunity is that the host does not need to make their own antibodies, but rather antibodies are given to the host. Okay, so someone else made the antibody, something else made the antibodies, and then you will receive the antibodies from that source. You did not make them. So as soon as you receive the antibody, as soon as these antibody goes into your system, you will be protected immediately. Immediate protection. But it will be short-lived, short-term. Because you won't be creating your own memory cells. So once those antibodies are broken down, the protection is gone. Are we all clear on the difference between active and passive? It just has to do with who's making the antibody. Are you making it? If you are, then it's active. If you are not, then it's passive. That's basically it, okay? So it's like, doing an assignment. If you cheat and you just copy someone, you get the answer right away, but you didn't learn anything, okay? So like any gain is short-term. But if you do your own assignment, it's not going to be done right away, but the benefit is long-lasting, okay? Maybe that helps. Now, under the active and the passive, we have two categories for each. It could be natural, or it could be artificial. Okay, natural or artificial. Okay. So an example of natural active immunity is it's an experience that we all have. Okay, so like we got sick from some bacteria, some virus, got us sick. Let's say you got the chicken pox, right? My favorite example. You got chicken pox, you got sick from it, okay? And then what happens is you recovered and now you are protected. Okay, so there is nothing unnatural about it. You caught the disease by natural means. Okay, there's no injection involved or whatever, right? You just catch it naturally, and then you got sick. You make the antibodies, you recover, and now you have memory cells, you're protected. Okay, but a lot of times we cannot afford to wait for the natural infection because some of the viruses out there are very virulent, right? We call them. That means that they can make you very sick in very short period of time. And if you wait for the natural immunity to kick in, right, during that whole time while you're waiting for the antibodies to be produced, the virus or the bacteria can cause a lot of harm, right? Which is why you might want to get vaccinated ahead of time, so that your body will be ready. So that's an example of artificial active. Okay, that is receiving a vaccination. So vaccination for for the flu, right? For polio, right? For COVID-19. These are all examples of artificial active. You are still the one who's making the antibodies, okay? But you are deliberately exposing yourself to these foreign antigens through the vaccination. That's why it's artificial, right? Okay, you're not catching it by natural means. And the whole point of the artificial active immunity is to get ahead of the game, right? To be prepared, right? To prime your body, to educate your body so that when the real thing comes, you will be protected. Does that make sense? Okay. Natural passive would be example of that would be like transfer of antibodies from mother to the baby through placenta. Okay, so antibody transferred through placenta. We know that can happen, right? We talked about that last time with the hemolytic disease of the newborn, with the Rh incompatibility. We know antibodies can go from the mother to the baby. But in this case, we're talking about protective antibodies, right? So if the mother has protection for the flu, right, or for the COVID-19, then her immunity can pass on to the baby through the placenta, right? And breastfeeding. But remember, these are passive immunity. So, as soon as the baby comes out, those antibodies that the baby got through the placenta will slowly break down, and eventually it will have no protection, right? So the mother can continue to breastfeed to transfer the immunity. But again, right, after the baby stopped breastfeeding, they would eventually lose the protection, and then they would have to get their own exposure, right? They could either get exposed naturally or through vaccination, right? And then they will make their own memory cells and get protected that way. Okay. And then the other form is artificial passive. So there are two types of artificial passive here, right? We can do the artificial passive for treatment. Okay, for treatment or for prevention. Okay, so an example of artificial passive immunization for treatment would be, let's say, you working at the hospital, and then you just give a shot to a person who has hepatitis, okay? And then you're supposed to discard the needle into the sharp container, but you accidentally poke yourself with it, right? So that's dangerous because you might potentially just expose yourself to hepatitis, right? So as a treatment, as a prophylactic treatment, right, you can receive a serum, right? A serum is purified antibody from people who are immune to the disease. Okay, sometimes they could grow these serum in animals, right? They could expose the animals to hepatitis, for example. They make the antibodies, and then you can purify it and create a serum from it, right? So basically, before the virus has a chance to replicate, you inject these antibodies and you protect yourself, right? I actually got bitten by a dog like recently when I was out for a run. And what happens is, like, in case the dog had rabies, then rabies can actually kill people. There's no cure for rabies. Okay, so I went to my family doctor, and even though we don't know if the dog has it, but as a prophylactic treatment, the doctor gave me serum. Okay, for the rabies virus, right? So I had to receive actually I received 3 shots on the first day when I went to the doctor, 2 shots for the rabies serum, and then they give me a shot for tetanus as well, because apparently, like, the dog's saliva can carry bacteria that could cause tetanus. So I got that shot as well, and then subsequently I have to go back every week for the whole month to keep on receiving the serum, just in case I caught the rabies virus. So that's an example of artificial passive. All right. So I hope you never got bitten by a dog, but there are other things that could give you rabies, right? What else that you encounter on the streets can give you rabies besides dogs? Anybody know? Yeah, raccoons, right? Okay, those pesky little things, right? They if they scratch you, if they bite on you, definitely could give you rabies if they're infected. Bats is another one, right? Sometimes people say they would find a bat in their house or something, right? Like, you should not try to catch them yourself, and you should not try to sleep with a bat in the house, right? Because, like, you never know when you're sleeping, they come down and bite you. You should get rid of it as soon as possible, because bats is they carry a bunch of diseases, including rabies. Okay, so that's for treatment. Okay, for prevention, here is an example for prevention. Okay, you would also receive a serum. Okay, serum after exposure, we would say after exposure. Okay, e.g., rabies, right? I said. This one, the prevention, the serum is if you have to go away for business trip, and where you're going has this prevalent disease, you don't have time to get your traditional vaccine, but you want to be protected. Okay, and so you would get a serum. Okay, it would give you temporary protection, and it will give it to you immediately. So when you go on your trip, you'll be protected. But next time, if you have to go again, right, you will have to get vaccinated again. The serum that you got the first time will not work. Same thing for me. If I get bitten by a dog again, right, next time I will have to go receive more serum. Okay, because the protection I got last time, which was almost a year ago, would not work anymore. All right, so there you have it. 4 types of immunity. Anybody have any questions, any comments, clarification that you need? No, I hope that all that makes sense. All right. Let's try some of these question. We're just gonna skip the break today, okay, and finish what we can. Joffrey is bitten by a rattlesnake while on a camping trip. His friends immediately rush him to an emergency facility. The venom is a protein-based toxin. What treatment would provide immediate protection, and why? So what should we give Joffrey here?
A serum.
A serum, that's right. So like an antitoxin, right? For the venom. But here's the tricky thing, right? Like some hospitals, they have a stock of these anti-venom, they call it, particularly in the States, right? Like if you think about places like states like Texas, for example, where there's a lot of desert, there might be a lot of like snake bites, and so they have a healthy stock of anti-venom. But in Ontario, you're not really likely going to be bitten by a snake, right? Unless you go purposely look for them and poke them with a stick or something. So there might not be an immediate serum that's available. And so people say, if you get bitten by a snake, if possible, you should take a picture of the snake so that they know what bit you, and they will be able to find the correct serum for you, quickly, right? Because sometimes you might have to request it from another place, right? most places do not have a complete stock of these anti-venom. So this type of immunity, right, explain what it is, that would be passive and it would be artificial, okay? If Joffrey were to be bitten by another rattlesnake in the future, will he be immune to the toxin? The answer is no, right? Just like I told you, these protections are just temporary. They are not for long term, and so you would need to have another serum. 1 dose of vaccine against chickenpox will virtually offer lifetime protection against the virus, whereas a new flu shot is needed every year. Explain the reason for this. Right? Have you ever wondered that, right? Like, you know, 1 vaccine for chickenpox, you're good for life, you know. Sometimes you get boosters, right? For like things like MMR, right? The mumps, measles, rubella, you might get boosters, but you know, once the boosters are done, you're protected for life. But how come we need new shots every year for the flu virus, right? And now they're saying we need new shots for the COVID, you know, every now and then, right? Anybody know why? Want to speculate on the reason? That's right, Mayfis, mutation. Prashil, you want to add to that, or I saw you unmute.
Sorry, Professor, I was about to tell that. Thank
You
you.
were about to say mutation as well. Yeah, exactly, right? And Mohammed said flu shot is short time. Well, you know, the mutation is a key piece. So let me just explain to you. By the way, does anybody know any flu on top of their head? Like you see them on the news. Sometimes they say, "Oh, watch out for this new strain of flu," and then they tell you the name of it. Does anybody know the name of any flu at all? If I say it, you would have heard of it for sure. But I just want to see if people are aware of any flu. Usually is some numbers and some letters to it. Anyone at all?
It's like bird flu.
Yeah, bird flu, right? Anybody or the avian flu, right? But the avian flu, it's like H5N1, right? Do you guys know what I'm talking about, right? Like you've heard that before, right? I'm sure H5N1. What's another one? There was actually another pandemic recently, right? Like in 2009, right? Does anybody remember the flu for the, yes, Ryan, H1N1, right? Okay, that's not the chicken one. The H1N1 is the swine flu, they call it. Okay, the avian flu, Ahmed, that's the H5N1, right? Not just chicken, but like birds in general, like including things like ducks and geese and whatnot. Okay, but you know what's the N and what's the H? What does it actually mean? Well, it turns out on the flu virus, the influenza virus, okay, on the surface, there are 2 antigens, okay? The 2 antigens are called hemagglutinin, which is what the H stands for, and neuraminidase, okay? So there are like 18 versions of the hemagglutinin and 11 versions of the neuraminidase. And if you do your math, 18 times 11, that's like a total of 198 different strains out there, right? Okay, and you know, every time they mutate, there's subtle changes in these H and N's, and sometimes the mutations will create a strain that is particularly deadly. Or if there are 2 strains, let's say there is a human strain that infects the pig, and then there is like a bird strain that infects the pig, the 2 strains can recombine with each other and create like a new strain, right? That's what the H1N1 was. We call these like recombinant viruses, right? And so, you know, every year the people who study to track these things, right? People at the World Health Organization, for example, they would predict what 3 dominant strains are going to be circulating that season, that flu season, and then they will make the vaccines for that. Okay, so those are only good for that particular year, and only if their predictions are accurate. Sometimes their predictions is like way off, and then you will hear people say, "Oh, this year's flu vaccine is only like 30% efficient." Right? We are a little bit fortunate to be in the northern hemisphere because we can look at countries in the southern hemisphere and see how their winter goes, right? And have an idea of how bad the flu season will be. Because Australia, right, they have the winter before us, right? Okay, and so we can look at the data and see whether the flu vaccines are actually, effective or not. So that's why you have to have a new flu shot every year, because the dominating strains are not the same every year, and there are subtle changes in these H and Ns that would cause the vaccine to become ineffective. So the goal of targeting flu and COVID really is to create something that will work for all kind of flus, right? Regardless of the mutation. You can think of the H and the N as kind of like this huge mushroom, right? On top of the flu virus, okay? And the cap of the mushroom is what's changing all the time, all these mutations. If you change the cap, then the antibody that worked before is not going to work anymore, okay? But the stalk of the mushroom, that's the part that actually doesn't change very much, okay? And so they're trying to create an antibody that will be able to recognize those portions of the flu. And so, it will be a universal vaccine, because regardless of how these are going to be changing in future years, if the stock remains unchanged, you will always be able to have an effective protection. Does that make sense? Okay. So same thing for the COVID, right? You hear all these variants, right? The Omicron strain, whatnot. It's the same concept. There are parts of the virus that keep on mutating, keeps on evolving. And if we're able to find a universal part that remains constant in all these strains, then we would theoretically be able to have one single universal COVID vaccine. Okay. That was a very long answer for this question, but I hope it makes sense. HIV virus, HIV, which is the human immunodeficiency virus that causes AIDS, infects macrophages and destroy them. Explain how this will affect your immune system. Anybody want to take a crack at that? If you destroy all the macrophages, what's going to happen to your immune system? Anybody?
That's why we are susceptible for other diseases, because no immune system.
Yeah, that's a good start. Yeah. And anyone else want to add to that? All right, so yeah, without the macrophage, your innate immunity will be basically weakened, right? You lacking all these phagocytes that would go around and mop up pathogens. So those are gone. But more importantly, remember the macrophage over here. Where is my drawing? The macrophage is responsible for antigen presentation, right? This is one of the cells that's responsible for activating the adaptive immunity, right? So without the macrophage, you have no way of activating the adaptive immunity. So people who have HIV, people who have AIDS, they could get very sick, very ill from common infections that we can fight off easily, right? So like you and I, if we get infected by a cold, that's okay. Like a couple of days later, we would feel just fine. But people with a we call them immunocompromised, right? Okay, they don't have the tools to fight off a common cold because they cannot activate the adaptive immunity, right? So basically, they have virtually no immune system, right? And so people with AIDS, they are immunocompromised, and they could die from simple infections. Okay, pneumonia is a common one that they could get, for example. I hope that answers the question. Now, Victoria says, "Is it going to attack itself?" That's for autoimmune diseases, right? That's when your immune cells is not able to distinguish what belongs to self and what belongs to outside body, right? The self versus non-self aspect is not there. In the lecture note, there is something called the tolerance test. I don't know if you guys get that far in the recordings. I mean, I do realize it's a lot of things to watch, but the tolerance test is the test that the T cells need to need to take before graduating. Okay, every T cells, remember, right? Each of the T cells has a unique receptors, right? Okay, we have to make sure those receptors is not something that would attack our own cells. Okay, so the tolerance test will check for autoreactivity. Okay, and you know, if it doesn't auto-react, that T cell is allowed to go to the thymus. But if there is autoreactivity, then we have to destroy that T cell. Okay, we cannot let that T cell come out. But you know, mistakes happen sometimes. We do create autoreactive T cells, and they end up attacking part of our body, right? So, for example, if we have T cells that attack the waxy coating around our neurons, then the person might get multiple sclerosis. Okay, if you have T cells that attacks your joint, you might get, you know, rheumatoid arthritis. So there are many different types of autoimmune diseases. Okay, last one. Many of the COVID-19 vaccines are mRNA vaccine. Based on your understanding of cell biology and immunity, speculate how mRNA vaccine works. I hope you know what APCs are now, right? The antigen presenting cell. I've only showed you one type of APC so far, right? Which is what? Just you know, tell me what it is. Just let me see if you're all with me. We only learned one type of APC. What is that?
Macrophage. That's
right, the macrophages, right? That's the APC. But there are other types of APC in the body, right? So there are these cells called the dendritic cells. Okay, they have long projections from the body. It looks like stars. That's why we call them dendritic cell. Okay, it's just another APC, right? So what happens is when you get your mRNA vaccine, the mRNAs are actually surrounded by little fat bubbles, okay, lipid bubbles, and these would help deliver the mRNA into the APCs. Okay? And as you know from last semester, you know, mRNA has the instruction to make the proteins, and in this case, that's the instruction to make the spike proteins on top of the COVID virus. Okay, and so the dendritic cells would translate the mRNA, and it would put the, you know, spike proteins on its surface. Okay. Right. What do we call this? Well, this is basically similar to the antigen processing, right? So now we would have a naive T cell with the right receptor. And if you don't have it right away, that's okay. By chance, you will make it in, you know, later on when you create more T cells. And that would activate the naive T cell. Then you will have clonal expansion, which ultimately would cause you to create plasma cells. And you would end up having a bunch of these antibodies that will recognize the spike proteins, right? So the spike proteins are non-pathogenic. They are not the part of the COVID virus that gives you the illness. Okay, they are just part of the structures on the shell, so they won't cause you to get sick. So when the real virus comes with the actual spikes and everything, along with the pathogenic part of the virus, you will be ready because you have all the antibodies. So some people usually ask me, right, like during this lesson, they're like, well, if we are not getting the actual virus, how come I get the fever? How come I sometimes, you know, get the chills? And you know, if you had the COVID vaccine, some of you might have very severe symptoms, right? Anybody had really bad symptoms from their shots, like first dose, second dose, very severe symptoms? Anyone at all? No? Yeah, it was just a walk in the park. That's good. Yeah, so for my kids, they had virtually no symptoms, okay? But for me, like I was out for 2 days for my first shot. Second and third was okay, but the first shot, my arm was super sore. I actually have a fever that was quite high, and you know, I was just exhausted. And so people wonder, right? Like, why? I thought you told me it's not the real virus. How come I'm feeling all these symptoms? Well, you have to understand, right? It's the vaccine is not the actual virus, true. However, you are still introducing foreign materials into your system, right? So anytime you introduce foreign material into your system, your innate immunity, right, all these mechanisms that we learned about is going to occur, right? You might have a bit of inflammation at the site of injection, for example. You're going to have the fever, right? And that's part of what happens when you introduce foreign substances. The key difference here between what is being injected and the real thing is the real thing contains the actual components that could make you very ill. Okay, whereas the vaccine, right, it's just the shell of the virus, so to speak, right, or the instruction to make part of the virus in this case, and that's not gonna kill you, right? It's not gonna make you very, very sick, right? The reaction that you get is basically just your innate immunity responding to a foreign substance. Okay, so I hope that makes sense. Okay. Let's do the fill in the blanks as well, and then we're gonna call it a day. Immunity recognizes PAMs, pathogen associated molecular patterns. Another name for adaptive immunity is acquired immunity, because you're not born with it. Okay, you're born with the potential to create it, but in order for you to actually get it up and running, you have to be exposed to the antigen, right? Microorganism that lives in or on us for mutualistic relationship. That means we both benefit from the relationship. That's normal flora. You know, some people are saying when you when you wash your hands too much with the sanitizer and everything, that actually kills off some of the normal flora on your surface. And you know, but not to worry, because they could come back. You know, some people are saying might be not too good of an idea to over sanitize, right? Because you're killing all these normal floras. Same thing. Sometimes you take antibiotics, right? Antibiotics kills bacteria. It doesn't know what's good and what's bad, so it kills off your good flora in the gut as well. And as a result, you might have a bit of a diarrhea when you're on your antibiotics. Okay, but you know, you can get them back. You can take probiotics, for example, or if you just wait, you know, your body will naturally replenish them. Perspiration, saliva, and tears contain antibacterial enzymes called lysozyme. Mast cells will release histamine. Macrophages can release colony stimulating factors. Right? These are chemical signals for calling for backup. Right? So the macrophage might not be able to clear the infection on its own, so you will release these colony stimulating factors, which will tell the bone marrow to make more white blood cells. Complement proteins are primarily produced in the liver. Complement proteins can assemble to form membrane attack complexes, which will insert holes in bacteria. The hypothalamus of the brain can increase body temperature, resulting in fever. Babies are born with anatomical barriers to disease. This is considered as innate immunity. You're born with it, right? Antibodies are Y-shaped proteins. It has a constant region located at the trunk and a variable region that is used to recognize antigen. Okay, so when you do gene shuffling, you are changing the variable region. This effector T cell will stop immune responses once the infection is clear. That will be the job of suppressor T cell. Naive T cells are activated by APC, such as macrophage. Naive B cells are activated by helper T cell. Antibodies are produced by plasma cells. Perforin are molecules used by cytotoxic T cells to form pores in target cell membrane. Right? They trigger apoptosis, right? Ronnie, age 6, has had chickenpox and recovered. He now has, what kind of immunity is that? Sure.
Passive?
Passive, because why? Did he make the antibody or did he not?
Well, he took a vaccine.
Oh, but this one did say vaccination, right? Just that he had chickenpox, right? That means he caught it, right?
Yes.
Yes. So in this case, it is natural.
Okay.
Yeah, and it's active, right? Because he recovered on his own, right? I hope that makes sense. Kathleen has been exposed to hepatitis. She will receive an injection of serum, which gives her. So this is artificial, right? Because it is an injection. Anytime you see injection, you inject anything, it's artificial. And this is passive. Right? The serum contains antibody, right? Sandy's going to summer camp. The physical form asks whether she has received immunization to measles and mumps. This type of immunization is, again, artificial. But active. Okay, she has to make her own antibodies. Baby Janice's mother has had mumps, which will give Janice natural, passive, right? Through breastfeeding, for example. Something is a severe allergic reaction characterized by systemic increase in histamine, that is anaphylactic shock. Right? It's like people with peanut allergy, right? The whole system goes into shock, right? And it's the blood pressure can get dangerously low because the histamine would cause the whole body to have leaky vessels, right? And the blood pressure can drop substantially, and that could be life-threatening. So what do we give to someone who is experiencing anaphylactic shock? Okay, what kind of jab do we give them? Anybody?
Right?
I'm sorry?
Steroid.
Yes, but do we have a name for that kind of steroid? I mean, it's a type of, okay, yeah, people are typing it, yeah. Epinephrine, that's right, which is a type of steroid, yes. Okay. The EpiPen, right? You might have heard that commercial, right? What is it? Blue to the sky and orange to the thigh, right? Tells you how to inject it. Something is an inherited disorder where a person is born without immune system, often called bubble baby syndrome. That is SCID, severe combined immunodeficiency. Okay. It's in your notes. Okay. So that is that. Let's do a few more polls. I still have quite a bit. And then we will end today. Go ahead. Okay, I am only hearing from about half of you, but I'm gonna stop the poll. Thank you for those who have participated. Okay, so which of the following are correct? Antibodies are Y-shaped protein. That's correct. Antibody produced against one pathogen will generally work against another pathogen. No, antibodies are highly specific, right? Okay. Sometimes in rare cases, it would work again for more than one pathogens, but usually not. Antibodies are produced by plasma cells. That's correct. Okay. Antibodies are found in blood plasma. That is also correct. We talked about that last time, right? When we do antibody for blood groups. Variations in antibodies arise from gene shuffling. That is also correct. Okay, so A, C, D, and E, they are all correct. Okay. Let's try another one here. Some hospitals, sorry. Of course, they don't do that anymore, but you know that was in the early days when people don't know what to do. That's one of the things that some people tried. Okay, so this is basically the same concept as the serum, right? So it will be artificial passive. Okay? First of all, you're injecting something that is taken from someone else. That is not natural. That's artificial. And it's passive because you didn't make the antibodies, right? Someone else did. Why do you need to receive a flu shot each year? We talked about this already. Because each year the dominant strain of flu virus circulating change. Okay, this one is a heavy one. So I'm going to give you a moment to try that. Go ahead. All right, I'm going to end that. And let's talk about it. So this is something that they actually do. They could take a T cell, take your naive T cells from your body. They could remove it, and then in a culture, they would basically expose them to cancer antigens, cancer cells antigens. And then once they differentiate, they would feed those back into your body. And so in order for this to work, all of the following must happen except for what? So number one, recognize an antigen that is found only on cancer cells and not other somatic cells. So that needs to happen. you don't want it to recognize something that's also on your somatic cells, right? Somatic cells are your bodily cells, right? So you need to be able to distinguish the two. I might ask, is this done in chemotherapy? No, chemotherapy is exclusively using chemicals to kill the cancer cell. This is called immunotherapy, okay? By tweaking your immune system to target the cancer cell is called immunotherapy. Okay, number 2, recognize an antigen that constantly mutates in cancer cell. So that's the exception. That's the answer. It's not possible. It's not possible to create an antibody that recognizes something that mutates constantly, right? The limitation of an antibody is that the strength and the limitation of the antibody is the same thing, right? Which is its specificity, right? So by having a high specificity, you can target precisely what you want to target and leave everything else alone, okay? But the downside of that is if the thing that you're targeting, right, changes because of mutation, then that antibody that you created will no longer be effective, right? Okay, so that's the exception, right? We're not going to be able to create a T cell with the receptors and therefore with the antibodies to constantly recognize something that mutates. Trigger clonal expansion, we need that, right? Because clonal expansion is creating a large pool of that effective cell, and it will become the effectors afterwards and produce effectors that will attack cancer cells. Of course, you want that to happen as well. So the answer is B. All right, just a couple of housekeeping items before we say goodbye. Next week we will be doing lecture 4, which is the last lecture before your test, right? So a couple things will happen next week. We will have the I will open up the lab, right? The lab quiz for you to do, and you actually should be working on your lab as well, because you need to do the lab before you do the lab quiz, right? So here, let me share my screen with you. Right here. So you see here, right? Next week, I will see you on Thursday again, and you will see that the lab quiz will become available on September 27th. Okay, you should not do it yet. Okay, some people do it right away as soon as they see it. But you know, you should wait until we have taken up the lab, right? In the announcement, I will post the link to the lab, which you will. It's basically the PowerPoint slides that you have to go through yourself, right? We will have time to take that up. Probably, you know, maybe the end of next class. Then you will be able to do the lab, right? So the lab is due on the October 4th. So you will have from the Tuesday to the Thursday to do it. And for the week number 5, we will actually meet on Tuesday instead of the regular Thursday, because Thursday you'll be taking your test, okay? And Tuesday we'll be doing the test review. Yes, the test one will only be on 2, 3, and 4, lecture 2, 3, and 4, okay? But the lab is for the whole unit, right? 1, 2, 3, and 4. Right. Like I said before, the lab, you will have 2 attempts and only the highest score will be recorded. All right. So yeah, that's it. Thank you for coming. And you know, we'll be in touch, and I'll see you next Thursday. All right. Have a good one. Bye now.